So it is Tuesday, and I'm going to review the homework assignments, do that fairly quickly, and then I can see if you guys have any additional questions. So if you have any additional questions on the Unit 1 stuff that you want to go over, and then I can start with the next batch of material. And remember, I'm planning for the first exam to be Thursday of next week.
Thursday of next week?
Yep, that sounds familiar, right? But yeah, so Thursday of next week. So that gives you plenty of time to go over stuff, and if you have some questions, you can, if you come up with some questions on Thursday, I'm going to move forward with the next stuff, but if you have questions, you can ask. All right, so the first homework assignment was pH, that sort of thing, so I'm pretty sure everybody ended up getting it all correct, but we'll go through it anyway. So a solution contains carbonic acid and bicarbonate. If you want to write out the equations or the chemical formulas, you could, or I'm just going to abbreviate. So HA will be the acid, A negative will be the bicarbonate, however you want to do it. Say the pK of the solution is 6.3. So the pKa for bicarbonate, carbonic acid is that. And we're going to say that the pH equals 7.2. First off, what's the ratio of base to acid? Okay, so first thing I'm doing is I'm reviewing the two homework assignments. So I think everybody ended up getting them all their points, but we'll go over it anyways. Okay, so I will give you constants, I will give you that information. I'm going to expect you to have some equations in your head. So one of the equations I'm going to expect you to have in your head is this Henderson-Hasselbalch equation for the weak acid, weak base. So by Thursday of next week, be able to write down pH equals pKa plus log base over acid. Okay, so commit that one to memory. Then we're just going to go through and solve. So there are 4 variables in this equation: pH, pK, concentration of base, concentration of acid. Now I'm only giving you 2, so we're gonna just consider that one variable the ratio. Are you with me? So I'm not going to ask you to solve that yet. We're going to take that in the second step. So for now, we're just going to solve it for A minus over HA. So 7.2 equals 6.3 plus log base over acid. I'm not going to bother drawing the brackets. Save myself some ink. Just subtract 6.3 from both sides. 0.9 equals log base over acid. And then remember the trick to get rid of the log, we take 10 to that power. Okay, so 10 to the 0.9 power will equal the concentration of base over the concentration of acid. And that ended up being 7.9. Okay, so 7.9 equals the concentration of base over the concentration of acid. And so there's 7.9 times more base than acid. By the way, one little check on it. Remember that the amount of base and acid will be equal. The amount of base and acid is equal when the pH equals the pK. So if the pH were 6.3, then they would be the same. They would be in equal amounts. The fact that the pH is higher than the pK means there's more base. So if the pH is higher than the pK, we're going to have more base. If the pH is lower, we're going to have more acid. And this is telling us that we got more base. So that's consistent. That's one way to sort of like check yourself. So the next part of the problem is Let's solve it. Well, I need more information. I would need more information in order to figure out how much I have. I can't solve an equation with two variables in it. So the additional information is to say that the total concentration is 10 millimolar. So if I tell you that the total concentration of the two is 10 millimolar, now by giving you an additional equation, we can go ahead and solve. So I can solve it for either term. It doesn't matter which one. I'm going to go ahead and solve this for the HA. I could do it the other way around. So let's solve it for HA equals 10 millimolar minus the base. I can then put that into the equation here. Before I do, I'll just solve this for the A minus. So the base equals 7.9 times the acid. And I can put the HA in for there. I could have solved this for the A, I could have solved it for the base and put that in. Doesn't matter, I'll get the same answer. 7.9 times 10 millimolar minus A minus. And that's going to equal 79 millimolar minus 7.9 base. And we remember, again, imagine I had the equation x equals 7.9 times y, or actually, hang on a minute, let me get this far here. Yeah, so A minus. If it helps you think of what we're doing here, instead of A minus, just think about it as like x. So x equals 79 millimolar minus 7.9x. So what do I got to do? I got to get rid of, I got to put my x's together. And over here, I've got 7.9x. Over here, I've just got 1x. So even though I'm not drawing a number there, that would be, that's 1, that's negative 7.9. Add 7.9 to both sides, and I get 8.9x. So 8.9 times the concentration of A equals 79 millimolar. And then we're just going to divide both sides by 8.9. The concentration of our base equals, what did that come out to be? That came out to be 8.8 something or whatever. I rounded it to 8. Yeah. I can, as long as you show your work, I can see where you rounded stuff. So if you're off a little bit here and there because of the rounding, that's okay. So, and the units here are millimolar. So the concentration of A, it might have been 8.8 something, whatever, blah, blah, blah. But there you go. And then the concentration of our acid just must be 10 minus that. So the concentration of the acid will be 1.1, approximately, depending on how you round it. Okay, so there you go. The other thing I like to do with this equation, I can't remember if there was one on the test last year or not. There might have been. We did an example at one point, I remember. But is where I give you some concentrations to start, and then I tell you, let's add a strong acid or add a strong base. Okay, so by the way, I'm just reviewing, so I just reviewed the first homework assignment about the pH, so I'm just doing review right now. So I'll leave that equation up there. See if you can do this one real quick. Let's suppose, hey, just doing some review. So let's suppose that our concentration of base is 5 millimolar, concentration of acid equals 7 millimolar. Let's just say, let's start off there. Let's say that it's carbonic acid again. So let's say that pKa equals 6.3. So do this for me, calculate the pH. So calculate the pH there. And that second homework assignment is due. If there's anybody that has it on paper, you can give it to me. If there's anybody who if you have submitted it electronically, that's fine. Did you change those numbers yet?
Yeah.
There you go. Okay. Oh, remember that you've got yours on paper. Thank you. Okay, so for this problem here, by the way, since we've just got one term over another term, one term up here, one term up here, it doesn't matter what units they are as long as you're consistent. So as long as you got the same units, it doesn't. So I could write out log 0.005 molar over 0.007 molar. I could write it out as molar. It doesn't matter. 005 over 007 will be the same as if I divide 5 by 7. So let's see. 5 divided by 7. And I did put them in the right places. Yeah. Okay, so 5 divided by 7 equals that. Take the log of that. Minus, I'm going to round it to minus 0.15. I get 0.147 something or whatever. If you want to write that out, that's fine. I'm just going to round it to 2 digits. So the pH of that solution is going to equal 6.3 minus 0.15. I think I can do that in my head. I think that gives me 6.15 or 6.2 if you wanted to round it to 6.2. Okay. So to be able to do that, that should be pretty straightforward. There might very well be a problem where you do that. I'm going to follow that up by saying, let's add the equivalent of 0.1 millimolar HCl. Okay, so let's say, so we're going to add the equivalent of, actually let's make it 1 millimolar, just for the heck of it, doesn't matter. But, so let's say we're going to add the equivalent to make 1 millimolar hydrochloric acid. So that means we're going to add 1 millimolar protons. We're going to make an assumption that every one of those protons will bind to a molecule of base. Okay, so if my base was bicarbonate, we're going to say that every one of those equivalents of H plus will combine with a molecule of bicarbonate, and that's going to give me a molecule of carbonic acid. So the proton will bind to whatever your base is, give you a molecule of acid. Technically, it won't be 100%, but it'll be like 99%, whatever, good, so we're not going to worry about that. So I'm basically, whatever the amount of the HCl is, I'm removing that amount of base, and I'm adding that amount of the acid. So if I add 1 millimolar of protons, I'm going to take away 1 millimolar of the base and turn it into 1 millimolar of the acid. So for every 1 millimolar of this, I subtract a millimolar of the base, and I'm adding a millimolar of the acid. Okay. Darius, so you're with me?
Yeah.
Okay. You had a look on your face for a moment. Maybe not. Okay. So now you just redo the math. So we take away from the base, we add it to the acid, and we just repeat the, so 4 divided by 8 is 0.5. The natural log of, the base 10 log of that is negative 0.3. So now the pH went down to 6. And by the way, I added acid. I said we're adding acid, so the pH should go down. The pH should decrease.
Oh, never mind. I did the LN by accident.
Oh, okay. I almost did, yeah, so. Okay, and then if I go back to the beginning, if I go back to the initial concentrations, 5/7, if instead of adding a strong acid, if I were to add a strong base, say 1 millimolar sodium hydroxide, now what you're going to do if you add OH is you're going to remove a proton, an equivalent amount of protons from the acid, and that will take away from the acid and give you more of the base. So in that case, the number of the concentration of the base increases, so that would go up to 6. The concentration of the acid would decrease by the equivalent amount. By the way, that one's an easy one to do in my head. 1 log of 1 is 0. That would just set the pH at 6.3. Okay, so what kind of things am I going to want you to do with this equation? If I give you concentrations of the acid and base and give you a pK, you should be able to calculate the pH. If I give you everything on the right side of the equation, you should be able to calculate the pH. Understand what happens if I add a strong acid or add a strong base. That's going to change the concentrations of the acid-base pair like we just did. If you add acid, you take away from the base, add to the acid. If you add base, you take away from the acid and add to the base. Yeah?
So I'm assuming if you made it like 2 millimoles instead, it would be up to 2 millimoles.
If I just made this,
Like the number itself, like it says 1. Yeah.
right? So if I could do any number here, I could say 2 millimolar. My numbers starting out were 5. My numbers starting out were 5 and 7. I could add 2 millimolar sodium hydroxide. What am I going to? I'm going to increase the amount of base by 2 millimolar now, and I'm going to subtract from the other. Yeah. Not to. Yeah.
Oh, just double checking. You said for the strong acid, you take away from the base and add as an acid.
When you add acid, you take away base and add to the acid. When you add base, you take away from the acid and convert it to base. And not to go too far with this, not to go too crazy, but I'm going to do one more. So what if instead of 1 millimolar, what if I did 0.1 millimolar? You know, feel free to use your calculators. This one I can do in my, as long as I have some simple numbers here, I can do it in my head, I think. But so now I'm adding base. So I'm going to add to the amount of base. Now that would just be 5.1.
Okay.
and I would subtract 0.1 from my 7 would give me 6.9. Yeah.
Oh, you added a base. Okay, got you.
Right. If I had said 0.1 molar HCl, this would have gone down, that would have gone up.
Yes, okay.
Would you ever give us when we have to convert the units, or would you, if you're asking a question like this, would you always put it on?
I am perfectly happy to do things where you have to convert units. I could have said 1 times 10 to the negative 4th molar. Darius is like, why? Why would you do that to me? So, I think I got that correct. I think 0.1 millimolar would be 1 times 10 to the negative 4th molar. Okay, so that's just those examples, the homework assignment, those things, that's what I want you to do with that equation. Looking at the second homework assignment. So I gave you an equilibrium constant. So I gave you a reaction, gave you an equilibrium constant. So we've got a reaction here, fructose 1,6 bisphosphate. That's a 6-carbon molecule, by the way. You didn't really need to know that, but FYI. And that can break apart into 2 other molecules, dihydroxyacetone phosphate, I'm going to abbreviate, plus glyceraldehyde 3-phosphate. Oops. And so I got a reaction here where the 1 molecule can break up into the other 2. I gave you an equilibrium constant for the reaction, 6.8 times 10 to the negative 5th. Okay, so calculate the standard, delta G. So here's another equation I'm going to expect you to stuff in your head. So stuff in your head, standard delta G equals negative RT times the log, natural log KEQ. Okay, and that natural log term came out for me to be negative 9.6. So if you take the natural log of that number there, you should get a negative 9.6. Multiply that by 2.48. Now, I converted it to kilojoules. If you had 2,477 or whatever, you could have something like that, joules per mole. That's fine. Okay, so whatever way, but I converted it, and we've got a negative times a negative. This is going to end up being a positive. So in kilojoules, I get 23.8 kilojoules per mole. If you had 23.8 times 10 to the 3rd joules, that would be fine. If you wrote it out 23,000, whatever, joules per mole, fine. And so one way or another, that's the standard, that's the first part. And then I say calculate the Gibbs energy. So the Gibbs energy is going to be the standard plus RT times the log, natural log with the equation, the concentrations written out. Concentrations of products over reactants. So I'm just abbreviating there. There's our numbers. And I think I said this a couple times before, I'll say it again. If the number of terms in the numerator is not the same as the number of terms in the denominator, you have to enter molar concentrations. Okay, so I think in the, at the end of the exam where I gave you the concentrations, I think I gave you some in millimolar and one of them in micromolar or something. I gave you in different units. In this case, you not only have to put them in the same unit, you have to put them in molar. So you're frowning. Okay, so yeah, you have to put them in molar. If there's 2 terms in the top and 2 terms in the bottom, it doesn't matter. As long as you're in the same unit, you could use millimolar, micromolar, but you have to put them in molar. So we're going to get, we already calculated our standard, 23.8 kilojoules per mole plus 2.48 kilojoules per mole. And the dihydroxyacetone phosphate is going to be 7 times 10 to the negative 5th molar. Glyceraldehyde 3-phosphate is going to be 5 times 10 to the negative 6th. Fructose is going to be 1 times 10 to the negative 4th. So I'm just in the habit of always putting these in molar concentrations. But if the 2 terms are not the same numbers in the numerator and denominator, you have to do it. That number came out to 3.5 times 10 to the negative 6. And the natural log of that number comes out to negative 12.6. Okay, so that gets you that far. Negative 12.6 times 2.48 ends up giving me, I get negative 31.2 kilojoules per mole. Again, if you have joules, that's fine, doesn't matter. And we're going to turn this into negative 7.4. Actually, that doesn't, I must have rounded somewhere else or something, but 23.8. Or no, yeah, 7.4 comes out right, kilojoules per mole. And again, you could be a little bit off of that depending on how you round it as you go through. But I would expect to see something around there. And I'm trying to remember if I said this or not. I know I talked about the near equilibrium reactions and the metabolically irreversible. So I don't remember if I said this or not. For a near equilibrium reaction, I'm going to say within plus or minus 10 kilojoules per mole. So anything between negative 10 kilojoules and plus 10 kilojoules per mole, anything in between that range, we would consider near equilibrium. Meaning that if the concentrations shift in a reasonable way, it could send the reaction the other direction. So, see if I can explain that first. So, this reaction is favorable under biological conditions. Under biological conditions, this reaction is favorable in the forward direction. Okay, so it's favorable for the fructose to break down. Because that number is within 10 kilojoules, if the reactions of these two things started to go up, we can get that reaction to start going in the other direction. We're not that far from equilibrium. So I may or may not go into this much when we talk about the glycolysis, but this is a reaction of glycolysis. This happens during glycolysis. Have you ever heard of a process called gluconeogenesis?
Kinda.
Maybe, maybe not. So that's the reverse. So glycolysis is taking glucose and turning it into pyruvate. You can take pyruvate and turn it back into glucose. So like after you exercise hard, you build up lactic acid. You can convert that lactic acid to pyruvate and turn it back into glucose and reuse that glucose. So when you're doing glycolysis, when you're breaking down the sugar, this reaction goes that way. After you've been exercising hard, after you've done some kind of workout or something, these two things are gonna build up, and you can actually run it backwards and make that and get the glucose back. So this reaction is designed to go in either direction depending on your circumstances. So if my muscles are working hard, this reaction's going forward. After I've worked out, if I've built up some lactic acid and I want to convert it back to glucose, we run it the other way. Now, back previously, I threw some numbers out at you to calculate the delta G for the breakdown of ATP. ATP breaks down into ADP plus phosphate, and when we went through some numbers, we came up with something like the delta G for that being something like negative 64 kilojoules per mole. That reaction ain't going backwards. That reaction is metabolically irreversible. So 64 is considerably larger than 10. That's way outside of that range. So I'm going to set the cutoff at about 10. So anything with a plus or minus less than 10, that's close enough to 0, that's close enough to equilibrium. Once we're outside of that 10 range, certainly something like 64, if the concentrations of ADP and phosphate start to go up some, that's not going to be anywhere near enough to make this reaction go backwards. You would have to have ridiculously high concentrations of these things that aren't ever going to happen in reality. All right, so that is that. That's it for the homework assignments. Anybody got any questions at this point? Okay, so yeah.
When it comes to the exams, and say like I use the calculator on my phone rather than to bring in a T9 for.
So everybody's going to need a calculator. I would prefer you not have your phone out when you're taking the exam. Or actually, let me not say I prefer, let me just say I don't want you sitting there with your phone. Okay, so bring in a calculator and your calculator has to be able to do like natural, the oddest thing your calculator probably has to do is a natural log. So make sure you've got a calculator that can do a natural log. Okay, if anybody, I assume everybody has a calculator, can get a calculator that can do that. If you have a problem with that, let me know. Okay, so we got 2 more class periods before the test. I don't want to spend those entire class periods reviewing. If it happened, whatever. I do want to move forward a little bit, but again, I'll give you time on Thursday of this week if you want to review. I'll give you some time Tuesday of next week if you want to review a little bit, to some degree if you got questions. Let's say that come to me with specific questions. Don't show up on Tuesday. Don't just show up here and say, "Can you go over everything?" I'm not going to go over everything, but try to between now and Tuesday figure out if there's something you need help with, and if you have specific questions, how do I do this? How do I use this equation? We'll review. So in the meantime, on to the next chapter, on to the next unit. So the next topic that's going to come up is amino acids and proteins. Any questions before I do that? Okay. All right, so.
Wait, can I take?
Oh,
No, it's okay, it's okay. It's okay.
Okay.
You're good.
I am recording,
Yeah.
by the way, so if you wanted to, you could go back and look over it. Yeah. I'm going to give the exam Thursday of next week. So in the syllabus, I think it just said like the week of February 16th. So the 16th is a Monday, so the week of the 16th is the Tuesday and Thursday, and we thought, well, everybody decided the further we can put it off, the better, I guess. So people voted for Thursday over Tuesday. So it'll be Thursday next week. All right, so the next unit is gonna largely focus on proteins. So we got, you remember your 4 classes of organic molecules. We got proteins, we got carbohydrates, we got lipids, we got nucleic acids. So we're gonna do proteins in a unit by itself. So we're gonna do proteins. Mixed in with that, we're gonna talk about some stuff about enzymes and how enzymes work. Carbohydrates and lipids are mostly used for energy, so we'll do carbohydrates and lipids in unit 3 and talk about how we get energy. We'll talk about glycolysis, and then we'll finish off unit 4, we'll talk about nucleic acids. So I've got a couple images to show to go along with the proteins and the amino acids. So, good news, I am not going to ask you to learn the structures of all the side groups of the amino acids. Okay, so I'm not going to go there. I don't think I can draw all 20 of them right now. I'm not going to ask you to do that. However, I'm going to expect you to know the basic structure of the amino acid, which should be pretty easy. So for example, if I were to tell you that why can't I think of one right now? So if I were to tell you that serine has the side chain CH2OH, if I were to tell you this is the side chain, if I give you the side chain, you should be able to draw me the structure of the amino acid, which means that you just need to know the basic part. And the name kind of tells you what it is. An amino acid, we have an amino group. So there's an amino group, and we have an acid group. Now an acid could be different things. In this case, it's a carboxylic acid. And in between, we got a alpha carbon, which will have a hydrogen attached. So there's our basic structure of an amino acid. I'm going to want you to memorize that. That should be pretty easy. And then whatever the side group is, you just add that in onto the alpha carbon. So remember that. And my little PowerPoint presentation starts off with that. So something that's a little pet peeve of mine, or something that I pay attention to at biological pH, at around pH 7, that amino group is going to have an extra proton. And so that will be an NH3 with a positive charge. At pH 7-ish, at biological pH, the carboxyl group will have lost a proton and be in that form. So it's pretty common to look up in books and see H2N COOH, and that is not the way it would ever appear. So kind of to refresh a little bit with some ideas about pHs and pKs. So the pK for the amino group is typically going to be somewhere around 11. So keep in mind pH equals pK plus log base over acid. See if you can figure this out. So if the pH is pH 7 and the pKa is 11,
Where you get 7 from?
I'm just making that up.
Okay,
So
okay.
I'm just getting that like approximate biological pH. What's that going to tell us about the ratio here? Our pH, if the pH were equal to the pK, if the pH were equal to the pK, what can you tell me about the amounts of these two things? They would be equal. So if the pH is much lower than the pK, which you're going to have more of? The acid. You're going to have much more of the acid. And so the acid in this case is H3 with an extra proton. The pK for the carboxyl group is typically somewhere around 3. Something like that. So the pK for the carboxyl group is going to be much less than the pH. So the pH is going to be much higher than the pK. That means you're going to have much more of the base. So that's going to lose its proton. So at biological pH, at a pH around 7, at a pH in your cells, in your body, the amino group is going to be in the acid form. The amino group is going to be in the protonated form. The carboxyl group is going to be deprotonated. So not to make a big deal, too big of a deal out of it, but if somebody writes it H2NCOOH, that molecule doesn't really exist. So what would I have to do in terms of the pH to have that in the H? I would have to be at a very high pH. I would have to be at a very high basic pH to get that in that form, and then that would take off that hydrogen. Anyways, let me shut up before I just confuse things more. Okay. So know the basic structure. I think when you look at, I've posted an exam 2 from last year, and I'm pretty sure one of the first questions is, here's the side group for such and such amino acid, draw the amino acid. So there you go. I want you to know that the different side chains fit into different groups. I want you to have an idea of that. Again, I'm not going to ask you to memorize any of these things, but we have some of the amino acids are said to be nonpolar. What are you going to see? You're going to see all CHs. So there's the amino, there's the acid, this is the side chain. And this is all just methyl groups here. Same thing here for valine. CH3, glycine just has a hydrogen. Proline here, it's got the one nitrogen group, which would be that's actually the amino. So proline has an odd structure in that the side chain actually comes around and attaches to the amino group. But that there, whenever you see that, that's just CH2, 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 CH3. So if the side chain is nothing but carbon hydrogen, it's going to be non-polar. Okay, so all of these, and again, I'm not going to ask you to memorize them. If I showed you a picture of leucine and said what category does it fall in, you should see that side chain is all just CHs, CH3s. That's going to be nonpolar. I crossed an X through this one. Some people consider that to be, or whoever made this picture considered that to be nonpolar, but usually we will consider that polar because the sulfur group is going to have polar bonds. Aromatic. So somewhere you might remember the word aromatic, that's where we have a ring structure. And this should be pretty obvious. So if I show you a side group with the ring, that should be fairly obvious that that's aromatic. I made a little note in the slideshow. This is where somebody got that wrong. They put the H2N and the COOH. They shouldn't have drawn it that way.
So how will you know if it's aromatic?
It's got a ring with a resonance structure to it. Do you remember where you have the double bonds there? Do you remember in some class somewhere about how those double bonds are going to spread across the entire ring?
Right.
That's called a resonance. So anytime the side group has one of these rings with those double bonds in it, that is, the word for that ring structure is aromatic. So an aromatic structure is that ring with the double bonds in it. Okay, so we've got nonpolar, we've got aromatic. By the way, FYI, these are going to be, does that look, how does that look in terms of being polar or nonpolar? Nonpolar, that's all carbon hydrogen. That's all carbon hydrogen, that's going to be nonpolar. That OH group is going to be a little bit polar, but most of this is nonpolar as well. I got the one nitrogen there, tiny bit, but again, most of that is nonpolar. So these fall under the category of aromatic. They're also going to be fairly non-polar. Okay, we've got some amino acids that are polar. The side chain is polar but not charged. So our amino acid is charged. Our amino acid is charged, the amino group and the carboxyl group, but the side chain here, so that OH, the OH group is going to be polar, OH is going to be polar, the SH is going to be polar, the amino group here and that carbonyl group, those will be polar. Same thing here. So these amino acids, you can see the CO, the CN, CO, OH, sulfur, oxygen, those are going to be polar. And then, oops, what happened to my charged? Oh, I got my slides out of, oh, no, okay, I remember why I put this out. Ignore that. All right. So, and then lastly, we've got amino acids that are charged at biological pH. So, aspartic acid, glutamic acid. What do we call the base form of aspartic acid? That would be called aspartate. So your asparagine is a different thing. So that would be aspartic acid. When it loses the actually, they wrote aspartic acid here. I'm just noticing they should have called it aspartate because it's lost its proton. So that's actually, if we put the H there, it's aspartic acid. Without it, that's aspartate. And glutamic acid, you've probably heard glutamate before. So glutamate and glutamic acid are the same thing, just with or without that proton. So those are the two acidic amino acids. And then we have lysine and arginine that will be positively charged. They pick up an extra proton. So the aspartic acid and glutamate lose their proton. The lysine and the arginine pick up a proton and they are charged. Will they be polar?
When they pick up an acid?
Well, when they're charged,
Okay.
that charge makes it very polar. So that the charge makes it polar, plus that CO is going to be, so these are going to be very polar, especially because of the charge. So what makes something polar is charge. If something has a full If an O has a full negative charge on it, that's very polar. If I have, so CO with the negative, that's going to be very polar. The C double bonded to an O, that'll have a partial negative charge on it. Okay, so know that there are different amino acids. Know the basic structure. I'm not going to ask you to memorize all those structures. Have that basic idea about the, some of them are polar, some of them are non-polar. I'm going to go back here for a moment. So, do you all have stuck in your head the word chiral? Okay, I'm pretty sure I remember learning about that or whatever back in the day. So, if you've got an atom that is bound to 4 different groups, it can be referred to as chiral, which means that if I made the mirror image of this structure, it would be a different structure. They would not be actually the same thing. So I don't know if I can draw that very effectively, but it's hard to draw in 3 dimensions. So they're showing here, we've got an alpha carbon, and they've got these 2 groups on the side are forward. So sometimes people draw it like this. You have the carbon. So these 2 groups are forward. And the other 2 groups, the R group and the H are set back. I should bring a prop or something here. It wouldn't be too hard for me to go buy some Kinder some toys or something, but whatever. So if there are 4 different groups, the mirror image of that molecule would have a different orientation. I don't know how to, I hope you understand what I'm talking about, because I don't know how I can explain it very well. One way to think about it, if we looked down, let's say we looked down on the R group, and then I would have an H, an H3N, and a CO. So imagine I'm looking down on it with the R up here somewhere. The mirror image of that molecule, H3N, HCO. If I turn that around, I can't, this is not the same orientation as that. So I can't turn it around and put them in the same molecule. Anyways, it's really not going to be that important that you understand that much. Otherwise, just understand that once we have 4 different groups, the molecule has this property where we said it's chiral. And depending on, there are 2 different forms. One of the forms is referred to as the L form, the other is referred to as D. Yeah.
What's that?
Okay, so, and I forget which is which, I get to, so there are 2, it's the same chemical formula, right? The molecules have the same chemical formula, it's the same stuff, but this one has a different structure than this one. This one has a different, it's put together in a slightly different way. Are you with me at all? Okay. So I could probably, somebody has decided, like, in the one orientation it's called, we're going to call that D, the other orientation we're going to call that L. Okay, where that comes from, this will probably not help understand all where that comes from. If I had a sample of D glycine or D, actually glycine would not be D or L because glycine, so if I had a D amino acid in water, when we shine light through it, it's gonna make the light or bend in a certain way. If we had the other form, it will bend the light differently. The light will be affected differently. And I apologize for not being able to give you a better answer to that, but the D stands for dextrorotary, and the L stands for levro, or something rotary. So it's whether there's a right hand bend to it or a left hand bend to it. So it's the same chemical structure, but for example, if I put this group here and the H over there, it's going to come out with a different orientation. We call one of those orientations D, we call the other orientation L. And and in biology, we pretty much always see the L form. So when your cells make an amino acid, your cells typically make the amino acid in the L form. So you're going to have like L-serine, L-methionine, L-tryptophan. For whatever reason, biological enzymes, biological systems typically just use the L form. If I can real quick find something else here. We've got words like stereoisomers and enantiomers. That doesn't really have a good picture of it. So again, I don't want to make too big of a deal out of this, but notice, so this is maybe it's a lot better than anything I can draw. So here are two amino acids. They have the same, assuming it's the same R group, they have the same structure. So I've got a carbon, I've got a carboxyl group, an amino group, a hydrogen, and an R group. These are mirror images of each other. Okay, so we got the NH3s to the front, the hydrogens to the back. I cannot take this molecule cannot be turned or manipulated to be in this same orientation.
So is it based on like the priorities of the other things that's connected? Like
Correct,
yeah.
yes. So I forget all the rules for that, but there's rules like the biggest group is like the number one priority or whatever and things, and I can't remember exactly how we determine which is the right, apparently this is the right hand form, that's the left hand form. I don't remember the rule as to, if like you grab it and with your right hand and your thumb points in a certain direction or something, then it's the right-handed form. I'm not gonna care about that. So all I want you to be aware of is that for all of the amino acids except glycine, glycine is not chiral, because for glycine, the R group is just an H. So if this is just an H, then I've got two groups that are an H. It's not chiral. I don't have four different groups anymore. But all of the other amino acids are chiral. The stereoisomers, there's a D and an L, and I want you to know biology typically uses the L. So for whatever reason, our bodies typically just use the L amino acids. Okay, where are we? 3 o'clock. Okay, so do a pair more here. I'm looking at page 2. So, and I'm going to get back to what I was talking about with the charges. So every amino acid has at least 2 titratable groups. When I say titratable, I mean a group that can gain or lose a proton. So our amino group can gain or lose a proton. The carboxyl group can gain or lose a proton. So there's a word for this particular structure here that comes up. An amino acid is a Zwitter ion. That simply means it has both positive and negative charges. And another word that applies is amphibolite. That means it can act as both an acid and a base. Amino acid, the amino part is basic, the acid part is acidic. Actually, they can both be kind of both. Right. So, consider this structure here. See, there's our alpha carbon, CO. I'm going to draw it this way. So, at a very low pH, at a pH below both of the pKs, how would this look different? If I added a bunch of acid, lowered the pH to a super low pH, something here is going to change. I'm probably not being clear enough about that, but how would this look different at a very low pH? The carboxyl group can pick up a proton. And so at a very low pH, both of these groups would be protonated. So at a very low pH, you protonate everything. Now, if this has a pK of around 3, and let's say this has a pK of around 11, by the way, this is how the course becomes cumulative, even though I don't go out of my way to make cumulative exams. I'm talking about pK, we're in unit 2, and I'm talking about pKs again. So it's kind of carrying over. So if I'm at pH 1, or pH 2, I'm below everybody's pK, everybody's protonated. Both of these groups are protonated. At pH 3, I would have equal amounts of this and that. Are you with me there? So at the pK, I would have 50% that, 50% that. As I go up above pH 3, I get more of the base. So above that pK, I've got more of the base. I've deprotonated it. As we raise the pH, I deprotonate that carboxyl group. Notice I'm still well below the pK for the amino group. So it's still holding on to its proton. At pH 7...
Quick question, right quick. So when you raise the pH to the carboxyl group above 3, what happens?
When you raise the pH, what is happening to the number of protons in solution?
It increases.
You had a 50-50 shot, you got it backwards. As I raise, lower pH means more H+. Lower pH means more H+. As I raise the pH, I'm getting rid of hydrogens. And what's going to happen to the hydrogens that are attached to, as I'm removing protons from solution, what's going to happen to these protons? They're going to come off the carboxyl group.
So they deprotonate.
So as I raise the pH, we deprotonate. And I get that this can be confusing, so I guess I get that. But imagine this does not have a proton. I'm going to take it from the other perspective. Imagine this does not have a proton. What happens if I start loading up? What happens if I start lowering the pH, loading it up with H+? It's going to be more likely for a proton to hop on there. What happens as I start taking away protons. As I start taking away protons, when that comes off, there's nothing to go back on. So low pH means more protons and more things are protonated. As we raise the pH, we're removing protons. This oxygen does not have a strong pull on the protons. This oxygen is pretty happy to let it go. Hey, you need to grab something? Oh, go for it, yep. So this carboxyl group, this oxygen, doesn't have a strong pull on those protons. So as we start raising the pH, it lets it go. Nothing?
No.
Something that sometimes happens, if something gets lost or something, sometimes they turn it in at the main office if you don't find it anywhere else. Okay, so the amino group has a much stronger pull on those protons. And I'll wrap it up in a minute. Yeah.
Oh, no, you can keep going.
Okay, did you have a question? Okay. I see a hand move sometimes, I think it's a click. All right, so the higher pK means that nitrogen has a stronger hold on those protons. But eventually, as I raise the pH, if the pH becomes 11, at pH 11, I'm going to have equal amounts of those two. And then as I keep raising the pH, keep pulling protons away, eventually it's going to let go of that proton, and I would have mostly H2N. So the amino acid can exist in different forms depending on the pH. At a very low pH, everybody's protonated. At a very high pH, they're both deprotonated. Because this has a stronger pull on a stronger attraction to the protons than this, in the middle, this will let it go, this will keep holding on. So at biological pH, around pH 7, this tends to have a positive charge, this tends to have a negative charge. And we'll talk, I'll finish that off, finish talking about amino acids on Thursday. And so, Sola gave me her homework, got Uche's, yep, so I'm good. So
I emailed it.
you emailed it to me? Okay. Do you want me to take a quick look at it? So, any questions before I wrap up? Okay. All right, I'm going to turn off the video. So.